by the time I'm done with my talk, you will know what this title means. So my name is Josh Barnett, as Gina has said, and I'm going to talk to you today about iron and sulfur cycling in an intertidal mixing zone of a coastal aquifer, with a particular interest to correlate how microbial communities in this environment affect the chemistry of the water. And what you're looking at here is our coastal aquifer of interest. You can see discharge channels caused by groundwater discharge that's going out to the ocean. And if you can see how they're dark coated with reduced iron sulfide during this month when this was taken. This was taken in July of last year. And in case you haven't had any chemistry courses recently, here's a little lecture on what I mean when I say redox cycling. Redox cycling in the environment has to do with reduction oxidation chemistry, which is concerned with the gain of electrons, which is reduction, and the loss of electrons, which is oxidation. And this is important for a lot of different chemical species in the environment, including iron. You can see in the oxidized version of iron, it's solid, whereas in the reduced phase of iron, it's actually in aqueous phase. And then you can also see the most oxidized phase of sulfur is sulfate, and the most reduced is sulfide. And these reactions can occur abiotically in the environment, but they can also be facilitated by microorganisms. So microbes don't just make you sick, they can actually have quite an effect on the environment. And what you're seeing here is an iron oxidizing microbe that is taking electrons from iron 2 in solution, and then it's precipitating out as this oxidized iron. So why is this environment important? Well, in this coastal estuary and in this intertidal mixing zone, you find something called submarine groundwater discharge. And this occurs in coastal environments when a coastal aquifer transporting fresh water from inland interacts with incoming seawater at the coast. And the submarine groundwater discharge is the combination of this fresh water and the mixing and recirculated seawater that's getting pushed out to the ocean. And in recent years, submarine groundwater discharge, or SGD, has been identified as having significant fluxes of nutrients and metals and other chemistry to the environment. It's actually been compared to river flow to the environment. And so because of this, it potentially has a large effect on global ocean chemistry. So these environments are really interesting to study, and one of our questions is, how do microbial processes contribute along these flow paths in this intertidal mixing zone? So here you can see, in contrast to the first picture I showed you, which the discharge was dominated by reduced iron sulfide, which was that black precipitate. Now you can see, and this was taken in a different month, that it's dominated by iron hydroxide, which is the oxidized version of iron, and that's indicated by this red-orange tinge. And now this is a good place to highlight why it's interesting to study iron in these environments. And that's because iron hydroxide, as you can see here, it's sticky. It grabs and sequesters other metals and nutrients from the environment, such as phosphate, lead, and copper. So if there's more iron hydroxide in the environment, then it can change the chemistry of the water around it, and maybe change the chemistry of the discharging water to the open ocean. As you can see here, we found that there were active iron oxidizing microbes at the surface of this environment. We showed that they were in this environment by taking samples and successfully cultivating them in the lab. This is a growth band of iron oxidizing microbes. So we showed that they're active at the surface, but what we wanted to know was if they're active deep within the aquifer, where most of the mixing between the fresh and seawater are occurring and most of the discharge is taking place. So other researchers have focused on these environments and looked more at the abiotic chemistry that's going on. And what they identified is something called an iron curtain, or a reactive iron barrier, that is often found at the mixing zone between the fresh water and the recirculating seawater. And this iron curtain is basically just an iron oxide and precipitated FeS-rich sediment layer that is usually placed directly at the point of discharge. So if you remember from the last slide when I told you how iron oxides are sticky, this iron barrier can have a huge effect on the chemistry of the submarine groundwater discharge that's being released to the ocean. So finding out more about the formation of the iron curtain, or the reactive iron barrier, will help inform about the chemistry going to the ocean. So one of our questions was, do iron and sulfur metabolizing microorganisms play a role in the formation of this reactive iron barrier? So in order to answer these questions, we went to Cape Henlopen, Delaware, which is part of the Delaware Bay. And Cape Henlopen in Delaware exhibits a dynamic intertidal mixing zone, as well as submarine groundwater discharge. And you can see, this picture was taken at low tide, the channels formed by the discharge that's going to the open ocean. And this dotted line is our sampling transect, which consisted of eight wells that went down into the subsurface. Each of these wells had 3 to 5 sampling ports per well, so it consisted of 33 sampling ports along this transect. And this transect, as you can see, is perpendicular to the coast and perpendicular to the coast. So this is our site, Cape Henlopen in Delaware. So here you can see some of our collaborators in the lab, or not the lab, the field, taking samples. Over here is one of the wells that I was talking about. It consists of Teflon tubing that goes down up to 4 meters in depth. And we use an electric pump to pump up the groundwater and then test it for the chemistry in the intertidal mixing zone using electrochemistry setups and this multimeter flow-through cell. Additionally, we were able to take samples for DNA extraction and microbial community analysis so that we could find out what microbes were present in the intertidal mixing zone. We also took some sediment cores of the top ½ meter at the beach in order to capture any particle-associated microbial communities. And you can see the visibly oxidized and the visibly reduced section of this sediment. Also, I pointed out a worm burrow here because whenever we would find one of these worm burrows, we would take samples for DNA extraction so that we could find out the microbial communities that are inhabiting these specific microbial niches. So our experimental approach, we went to Cape Henlopen, Delaware on a monthly basis last summer from June to October. And like I showed you in the previous slide, we pumped up pore water and took samples from the sediment. And what these samples gave us was an understanding of the chemistry in the intertidal mixing zone, and spatially where certain chemicals like iron and sulfide are within this zone. And then we were able to extract DNA and do microbial community analysis to find the predominant microbes in the intertidal mixing zone. And then once we knew the predominant community members, we were able to predict the microbial metabolic potential of these communities. So this allowed us to correlate the microbial community structure and function with actual changes in chemistry and changes to the iron barrier, or the iron curtain. So here you see our salinity profiles from 3 months of sampling. So salinity is basically just how salty the water is. So by taking our salinity profiles and contouring them, you see a cross-sectional view along our transect, which is the same view as this schematic here. And you can see that by utilizing salinity as a conservative tracer of the flow paths in the system, you can see salt water coming in through the top and making this flow path 1, and then the aquifer with the fresh water coming in and discharging right here. So in these 3 months, the flow paths did change like relatively a lot. You can see in August that the upper salinity cell was compressed much further down into the aquifer than the previous months. And this is because we think a change in freshwater flux coming from the aquifer. So what does this change in flow paths mean for the microbes? Well, we don't expect that the microbial niches will vary widely by month, but we do think that these changing flow paths may be able to disrupt the balance of iron and sulfur phases within the intertidal mixing zone and renew nutrient resources, so hopefully these microbial communities will be able to thrive in this environment. So now I will focus in on those reactions that are important to iron and sulfur cycling in the intertidal mixing zone and impact the reactive iron barrier. I'll focus on data from July because I don't want to go on for hours. But as you can see here, this is the black lines is just an overlay of our salinity, just to orient you to what we're looking at. This blue line is the beach surface. These dots are our sampling ports along our transect. And then these 3 red paths are 3 main flow paths throughout the system. Flow path 1 is intruding seawater coming in from the tide and from the waves. Flow path 2 follows the fresh water coming in from the inland aquifer and then discharging out from the discharge zone. And then flow path 3 shows the recirculating seawater coming in from the ocean. And it follows this schematic as well. So now, this is our oxygen and nitrate data from July. You can see that oxygen is represented in blue and nitrate in green, with more concentrated values as darker colors. You can see that oxygen and nitrate are primarily located at the top of our system, near the surface, and that's because they're being brought into the system from that flow path 1, the upper saline cell. So now you can see our iron and sulfur data with oxidized iron in orange and reduced iron in red, and sulfide in yellow. So you can see that iron and sulfur is predominantly located along the right side of our transect, or what I will call the offshore interface. So now I'll identify reactions that are critical to this iron and sulfur cycling that make this reactive iron barrier. So along flow path 1, coming in the ocean water, comes organic carbon, nitrogen, oxygen, and sulfate. And you can see this, it's called a geochemical zonation here, where oxygen is up at the top, and then that gets depleted, and then nitrate, and then sulfate, which is probably here, but we only measured for sulfide, so you just have to see the absence of sulfate there. And through our DNA analysis of the microbial communities, we've been able to implicate some organisms with causing this zonation. So up at the top here, you can see in this pie chart that diatoms make up approximately more than 50% of the microbial community in the surface. And diatoms are primary producers, so they can produce organic carbon. And then Bacteroidetes, which is an aerobic organotrophic microbe. So what that does is it oxidizes the organic carbon, similarly to what we do. We oxidize organic carbon. And then it reduces the oxygen to water. And that's why you see the oxygen being depleted pretty close to the surface. And then as we keep going further, we get nitrate reduction, and then important for the reactive iron barrier, we get sulfate reduction by sulfate-reducing bacteria. And we found in this portion of the flow path a predominant community member, which is a sulfate-reducing bacteria called desulfobacterales. And up here you can see some of the reactions that these microbes are facilitating, such as desulfobacterales is reducing this sulfate to sulfide. And we see the presence of sulfide here as a result of that. These are just some acronyms that I'll be using for these microbes. So now moving on to flow path 2, coming in through the inland aquifer, this water is very low in oxygen and has trace amounts of iron. We think that this is the source of iron to our reactive iron barrier. So by looking at the spatial location of the iron sulfur species, we can predict the extent of the reactive iron barrier. And this is consistent with what the other researchers have found, is that this reactive iron barrier is often found where these 3 flow paths meet and right before discharge to the ocean. So now I'll focus in on this critical iron and sulfur cycling portion of the intertidal mixing zone. So coming in along flow path 3, similarly to flow path 1 because it's from the same source, the ocean, it's rich in sulfate and organic carbon. So it comes in, and the organic carbon is oxidized. There's a lot of stuff in this slide, but I'll get through it all. It's oxidized by organotrophic bacteria and iron-producing bacteria. And we found Geobacter, which is an iron-reducing microbe, made up a predominant member of the community here at depth along flow path 3. So we see iron 2 here, and that's probably a result of Geobacter reducing iron 3 to iron 2. Also along flow path 2, we find another desulfobacterales driven community, which is a sulfate-reducing microbe, and you see this is why we see sulfide down here in this section of the intertidal mixing zone. And then as we progress up throughout the zone and closer to the site of discharge, we find an iron oxidizing community made up of an organism called Maripor Thiobacillus. And what this does is it takes the iron 2 and oxidizes it to iron 3, and that could have been why we saw so much iron oxides at the site of discharge during one of these months. And then we also found Sulfuramonas, which is a sulfur oxidizing microbe. So it'll take this sulfide and oxidize it back to sulfate, and this is why we don't see sulfide anymore in this portion of the flow path. So what this tells us is that this mixing of active iron and sulfur species enables many microbial niches for these types of microbes in this environment. And that they're located spatially so that they're implicated in the formation of this reactive iron barrier, which then this reactive iron barrier can sequester other nutrients and change the chemistry of discharging water to the open ocean. So although I'm not going to go into depth into this topic, we found that in worm burrows throughout this environment, a highly enriched community in the iron oxidizing zeta proteobacteria, up to 24%. And we think this is because these worm burrows allow greater oxygen diffusion further down into the sediment for these oxygen-dependent iron oxidizers. So in conclusion, this intertidal mixing zone brings together organic carbon, oxygen, iron, and sulfur, and it creates a mixing zone and niches for organotrophs, sulfur metabolizing, and iron metabolizing microbes. And we found these microbes situated spatially so that we think they're forming this reactive iron barrier, which then has the ability to sequester other nutrients and metals from going to discharge to the ocean. And then, as you can see in this schematic, some of these microbial processes are going in different directions. So the extent of the iron barrier depends on the relative rate of microbe metabolism. And we can't say for certain whether they're actively cycling yet because we did DNA-based analysis, so that just tells us that they are definitely there. So we're kind of predicting what kind of like metabolic reactions they're facilitating. But they're definitely implicated in the formation of the reactive iron barrier. So future